You've got some residual carbon-nitrogen substrates, which is really important when you think about the sort of ageability of your wine, how stable it'll be over a long period of time, and some trace elements and compounds. And so the interesting thing is, can you take, knowing all of the various components of wine, can you take water, add alcohol, add tannins, add various types of compounds that you know are present in wine, and get back wine? And the answer is no. You absolutely can't. I think someone's figured out how to now powder alcohol, so you can get like a powder where you can rehydrate alcohol and take it camping or do something else with it, but you can't do that with wine. It's just not going to work, and I don't really think there's ever going to be a point where we have the technology to reconstitute wine. At least not real wine. You could probably make some really gross tasting thing. Anyhow, what I want to get at here is everything in wine is matrix dependent. So this is something that our Hildegard Heyman, who runs the sensory lab in our department, talks about constantly. And idea is wine is a matrix, and what that means is all of those components that are in there are all interacting with each other. So there's probably, in the wine you guys are drinking right now, there's probably something that is, you're smelling it, but it's the way you're smelling that compound is being influenced by the presence of other compounds that you may not smell. So it may be masking, it may be enhancing. There's a really insane way that different compounds will layer with each other to help you experience a particular ester or something in a way. So those are the sort of matrix effects of wine. And we're going to kind of keep rolling here on talking about wine processing. So really big fundamental differences in wine between white wines and red wines is generally white wines are brought into the winery, immediately destemmed and put into a crusher, and the juice is taken off and fermented relatively cool. Red wines, on the other hand, are brought in, occasionally processed off the stems, sometimes they're left on the stems as well, and then they're macerated. So that's the guys tromping around in the tank. You pretty much don't see that at any modern winery ever anymore. That's not really a thing. Occasionally, yeah. Usually there's an intern who has a big stick who has to go and just kind of like mash this stuff down. But machines do a lot of the work. And this maceration process is part of what's imparting the depth of color in the wine red wines. Whereas white wines, because they're taken off the skins almost immediately, don't get that depth of color. Occasionally you'll see something called an orange wine, which is basically a white wine made with skin contact. They're weird. You can see, once you try one, you're like, I see why they don't really make wines like this all that often. Anyhow, so after maceration, then the red will go into a press, just like a white would. What we use these days is a big bladder press usually, so it's sort of a big chamber with essentially a giant balloon that you can blow up to specific pressures, deflate, blow it up again. What you're getting is sort of even pressure across all the berries, trying to extract as much juice as you can without extracting tannins from the seeds, because seed tannins tend to be relatively bitter. But again, what I want you guys to sort of see from this relatively complicated slide is there's a lot of choice points in winemaking. There's a lot of different points where you make, as a winemaker, you make a decision about your wine, and that influences the way it's going to taste, the way it's going to age, the style of wine you're making, and potentially even the robustness of your fermentation, which is going to be really important as I go on into my research, because that's kind of what I work on. So some introduction to some of the basic microbes in wine. Saccharomyces cerevisiae, or yeast, is your big player. Hopefully it's something you guys are all relatively familiar with. It makes bread, it makes beer, it makes sake, it makes most fermented beverages. Saccharomyces is a very important, very complex and interesting organism. When the grapes come into your winery, there's often some other non-Saccharomyces yeasts on there, stuff like Clechera, Hansenia spora, other things that don't do a whole lot of the fermentation, but they do tend to contribute some aroma characteristics and some complexity to your wine. They can also compete with the yeast for nutrients, which is important when we talk about dominance. The other types of things you see are some of these, there's lots and lots of bacteria, but these are sort of the relevant bacteria, are the lactic acid bacteria and acetic acid bacteria. So lactic acid bacteria, they make lactic acid. Acetic acid make acetic acid. Acetic acid bacteria are interesting because they're basically what turns your wine to vinegar. So if you've ever had a bottle that was open for a couple of days and you taste it and you're like, man, this is really vinegary, I'm going to cook with this now instead of drinking it, it's because acetic acid is pretty omnipresent, ubiquitous in any wine, anywhere, and as long as you give it a little bit of oxygen, it can start transforming ethanol into acetic acid as a way of making energy. So alcoholic fermentation, relatively simplistic. Yeast takes sugar. In wine, that's a mixture, 50-50 mixture of glucose and fructose that's coming out of the grapes. It's going to transform about 95% of that carbon into ethanol and CO2. About 3 to 4% of that will go to biomass, and about 1% will go to other metabolic byproducts, which is part of what you're smelling when you drink wine. But there's sort of a question of why do fermentation? And part of it is because yeast can gain an advantage by doing fermentation. Yeast are incredibly dominant in fermentation, and this is kind of a complicated graph, but really what all you guys have to see is when grapes come in and they go through the press and you put your juice into a fermenter, there's a high level of non-Saccharomyces yeasts, and they'll die off relatively quickly. As soon as they build up to about 4 or 5% alcohol, all those non-Saccharomyces yeast die. The Saccharomyces yeast take a little bit of time to get going. They'll go through sort of exponential growth. They'll hit a stationary phase. Most of fermentation happens during stationary phase, so you see a drop in Brix here. Brix is a density measurement that is essentially equivalent to 2% sugar. So Brix is a measurement that winemakers use to track loss of sugar and progress of fermentation. As the yeast go through stationary phase, they've sort of solidified their membranes, and they're starting to just run fermentation as a way of producing ethanol and gaining back energy. Eventually, once fermentation finishes and they run out of sugar, the yeast will start to die. And this is very important in traditional winemaking because the yeast will start essentially releasing their guts into the wine, and yeast guts are really tasty. If you've ever drinking champagne, that is the flavor of yeast guts. All that nice kind of bready flavor is just from yeast guts. It's also really important when you think about traditional winemaking because most reds undergo something called malolactic fermentation, and that's where all the malic acid is converted into lactic acid, which is a slightly softer acid. It's a little bit rounder. It's not quite as sour, and that's done by lactic acid bacteria, one in particular called Enococcus. And Enococcus cannot grow in wine until yeast release some of the nutrients that they've been holding on to. So this is sort of it's an ecological succession of different organisms to make a product. And here in this phase, there can be a lot of different bacteria that get going, sometimes for better, sometimes for worse. So frequently, winemakers will add SO2 or sulfur dioxide at the end of fermentations to really clamp down on the amount of microbial activity before bottling, because you generally do not want microbial activity in the bottle. You know, if you have a beautiful wine in your winery and you throw it in a bottle and you try it a year later and it's spritzy and weird and strange, that means it was not ready, it was not filtered properly, it was not treated properly, and you had microbial growth and instability within the bottle. So, why are yeast doing fermentation? If you think about it, if they do oxidative metabolism, they can get about 32 ATP per sugar. And ATP is essentially energy money that the cell can use to do work. So wouldn't you want more money rather than less money? That's a simple one. But that's not what yeast do. If yeast have sugar, specifically glucose, they say, man, I love glucose. All I can do is think about glucose all the time. They are literally obsessed with glucose. And when they have glucose, they'll do fermentation, whether or not there's oxygen. In fact, even in the presence of oxygen, they'll do fermentation. And so it's an effect called glucose-associated repression of alternative carbon sources, which is kind of a mouthful. So I'll just say glucose repression. But essentially it means that they're obsessed with glucose, and they'll keep using glucose until they run out of glucose, and they'll keep doing fermentation. Fermentation, you're only getting 2 ATP per sugar. Why do they do that? Well, they can make alcohol, and it gives them a competitive advantage. So yeast are incredibly dominant in these fermentations. They're able to come in, I'll go back and show you guys that you start out with incredibly high populations of non-Saccharomyces yeast, high populations of bacteria. Yeast grow up, dominate everybody else, and kill virtually everyone else up until they start dying off and something else starts going. So they're incredibly dominant in fermentation. And that's because of that glucose repression effect, right? They can, when they're so obsessed with glucose, they can take their time just doing fermentation, spitting out alcohol, and giving themselves an advantage. Some of the other things that make them incredibly dominant is the kinetics of their oxygen uptake. So they uptake oxygen very, very quickly, and all of the other organisms in there need oxygen to run their metabolism because they're not doing fermentation. They deplete the nutrients, or the macronutrients, so all the sort of amino acids and some of the micronutrients, stuff like biotin and vitamins and cofactors. They have high ethanol resistance. Most organisms cannot hang out and live at like 10, 12% alcohol, but most yeast, they do all right. They're not doing great, but they do all right. And yeast also have tolerance to sulfur dioxide, one of the tools that winemakers use to control microbes in fermentation. They have tolerance to hydrogen sulfide, which is something that a gas that yeast spit out as a way of sort of talking to each other. It's a quorum sensor. It's also, if you've ever opened a brand new bottle of wine, it smells like farts. That's H2S. It will usually blow off. We have an incredibly low threshold for it, so we can detect it very easily, but it's very, very volatile, so it'll blow off pretty quickly, which is why if you swirl a glass of wine, it tastes better. And they're also pretty resistant to other types of toxic compounds that can be present. So I spent a while Googling yeast cartoons for this presentation, and I like this one. Anyhow, so for the most part, yeast makes great wine, and it's not usually an issue, but sometimes it is. And this is what my lab works on, and what my boss, Linda Basson, has been doing for 30-something years here at UC Davis, has been looking at problems with fermentation, so causes of arrested or sluggish or just general problem fermentations. This is, again, sort of a Brix graph, so this is a sugar loss graph. This is just reduction in percent sugar over time, basically. The red one represents sort of a relatively normal fermentation where you don't have problems. By the time you get into negative Brix, you've got a ton of alcohol, and your wine is going to be relatively stable as long as you treat it right and keep oxygen away from it. But all of these other black lines represent different types of problems that can happen in fermentation, and most of them are going to be based on the yeast being stressed out or unhealthy. So what are some of the causes of stuck fermentations? They've been incredibly well documented over the years, but there's still some things we don't entirely understand and reasons why fermentations stick that we can't fully explain. So some of the sort of things that we do know are nutrient deficiencies or imbalances. So this can be stuff coming in with wrong set of amino acids in fermentation, and yeast are never really able to get going. You can suffer temperature extremes. In a really, really big tank, fermentation produces a lot of heat, so you can get bubbles of like extreme heat that can actually kill the yeast. Presence of inhibitory or toxic compounds, so sometimes bacteria can produce enough of some of the acetic acid or lactic acid to actually inhibit the yeast. Poor fermentation management is a big one. This again kind of goes a combo of all the things we were just talking about. If you have a huge tank and you're not mixing it enough, you can get those temperature gradients that can kill yeast. Not checking the nutrients in your wine and adding the appropriate nutrients before you dump the yeast can cause problems. So a lot of this comes down to things that you can actually do something about if you're a mindful winemaker. Again, inappropriate strain selection is one that, as a winemaker, you should really be having a sense of what you're doing. If you know you have Lodi Zin, which is like 26, 27% or Brix, and that's going to make a wine that's like 16% alcohol, don't choose a wine that can only stand 12% alcohol because you're going to run into a problem. And my favorite one is microbial competition, which again kind of goes back to some of the nutrient and balance stuff, because microbes can get in there, grow a whole bunch before the yeast goes, and screw up the nutrient composition and also release some of those inhibitory compounds. Point is, all of these things cause stress in these poor little yeast. I have so much sympathy for these guys, especially because I've killed so many of them over my years here. Anyhow, so how do yeast deal with stress? Yeast deal with it by adaptation. And I warn you, the next slide I'm going to show you is very complicated and very busy. Don't be scared. It's really just to drive home a point that yeast have a lot of different ways of adapting to stress. As a human being, if we were sitting out in the rain and it started raining on us, what would you do? Yeah, go inside, get an umbrella, find an overhang, something. You wouldn't sit there and just be rained on. Yeast don't have that option, right? They're not modal. They're stuck in their environment, so there's nothing that they can do. They might start restructuring their skin as a way of resisting the rain, we can't do that. They can. Anyhow, so this is an overview of the stress response pathways in yeast. And again, complicated, but what I really want to drive home to you guys is just there's a whole bunch of different pathways. So each one of these sort of little acronyms represents a different pathway. And what happens when yeast experiences stress is they're either adapting through changing expression within the cell, or they're activating, and part of that is activating molecular chaperones. So these are little proteins that go in and help refold other proteins. And this is helping them adapt to the stressful conditions because the outside environment is putting a tax on the inside environment. And what's really important for yeast is to maintain the homeostasis of their insides, right? If they lose inside homeostasis, they die. So they'll do virtually anything they can to maintain sort of the internal integrity of their environment and keep that different from the outside environment because that's what allows them to sort of do work and make energy. So one of the big things that can happen when you throw off your internal homeostasis is the yeast kind of go into like a scramble mode, right? And this is full on like we've just lost, the roof flew off the building and we're not sure what to do, so we're gonna activate prions. So you guys might have heard of prions before in the context of disease, mad cow disease, Kuru, scrapie, this kind of stuff. Misfolded proteins that get into your body and smash through your brain, causing renegade protein folding and all sorts of terrible neurodegenerative diseases. This is not the same thing. Molecularly, they are related. And in fact, we're sort of hoping to find a different term other than prion to describe this phenomenon. But for the moment, these have all the genetic characteristics of a prion. So prions, in some ways, we're going to step back from the disease model and think about just what is a prion. Prions are proteins. And they're generally proteins that are capable of two stable states, one of which is a self-templating heritable state. So this is changing the central dogma of biology. Central dogma says DNA makes RNA makes protein, and that gives you your phenotype. We're talking about a system, we're going like back to pre-1950, before we knew about DNA, where people still thought protein might be the heritable genetic information. And I'm telling you that you can actually inherit genetic information from generation to generation based on a particular protein fold. So the idea here is that an environmental stress has caused a chaperone protein to refold a protein within the cell. And generally these are proteins that have an unstructured domain. So say there's like a wavy arm of your protein and you experience the stress, the chaperone protein comes by and suddenly structures this arm into a particular conformation. And this conformation is then both self-templating, so the other proteins that have the sort of wavy conformation will go into the structured conformation, and that structured conformation can then be transferred from generation to generation. So it's a form of epigenetic learning that happens within yeast cells. And so there's been, I think 35 something prions that have been pretty well characterized, and then there's a whole other class of new ones that we're just discovering, and I've been working on one of those new ones that's sort of exciting, and I will tell you about that one. So first, I'm going to go back to just fermentation. Yeast loves glucose. They really, really, really love glucose. This is what facilitates fermentation. They make ethanol, CO2, dominate your competition. The reason they can do this is because about 50% of the protein in their membrane is this ATPase PMA1. So this protein just hydrolyzes ATP into ADP using energy money and shoots protons out of the cell. And that's really important because protons, when they build up inside of the cell, would throw off the homeostasis of the cell and would sort of shut down the yeast metabolism. And eventually, in a wine, what kills the yeast is the membranes become leaky, and the ethanol and the protons will start seeping back in, and the yeast will die. So pumping those protons out is essentially the thing that keeps the yeast alive. My prion, known as the GAR prion, which means resistant to glucose-associated repression. You can see hopefully where this is going. This is a situation in a lab where we give the yeast something that looks a lot like glucose, so it activates their glucose repression response, and they say, oh yeah, we got glucose, let's eat it. And then they get stuck, because it's like chewing on plastic food. You're not going to get anything out of it. And they have to learn how to circumvent this glucose-associated repression response and use this alternative carbon source. And so this happens at sort of a relatively predictable frequency within a particular strain. What's really interesting is that wine strains induce this at a much higher frequency than, say, a lab strain or a beer strain. So any yeast that lives sort of in isolation is generally not very good at inducing this particular prion, but yeasts that see lots of stuff in their environment, like wine yeast, fruit yeast, soil isolates, are all fairly good at inducing this prion. And what you get is in this unstructured domain of PMA1, that proton pump, you get a conformational change here, and this gives you this phenotype of the ability to grow on alternative carbon sources in the presence of glucose. And this might not seem like that big a deal to someone who's not a yeast biologist, but functionally, what it does is it can screw up fermentation. So winemakers started to care about this. I was working on this stuff, and I figured out that these GAR plus cells were not as good at fermenting. And this became really interesting. We started wondering, hey, is the establishment of this GAR plus prion something that could be affecting fermentations in the wine industry, or something that we've been seeing? So they can circumvent glucose repression, which is an interesting phenotype. It mimics a mutant phenotype, but the cool thing about these prions is that they're not a permanent change. So a population can induce a prion as a little like switch flicking kind of thing, but it's not permanent. It's not a mutation, so it's not in their genome. So after a couple generations, if the pressure leaves, they can uninduce that prion, and so it's not a permanent change. It's kind of like quick learning. There's also, you get downregulation of a particular sugar transporter, so this, again, relates to why they would be bad at fermentation. And we've seen that a GAR plus cell has sort of reduced fermentation capacity, so that's both alcohol production as well as microbial dominance. So here's a little sort of graphic. This is from a paper we published in Cell back in 2014. So the idea that there's bacteria present in fermentation. They're spitting out sort of a small molecule effector that the yeast are perceiving as a stress signal. Yeast decide to sort of fundamentally change the way that they interact with their environment by changing the conformation of PMA1, and that results in the induction of the GAR plus prion within this yeast population. And that ends with reduced alcohol and sort of slower fermentation kinetics and far more bacteria being viable during fermentation. So this is not what a winemaker would want for the most part. And it's been really interesting too, because a lot of different types of bacteria can induce this prion, but very specifically some of the wine spoilage bacteria, some of the lactic acid bacteria, some of the acetic acid bacteria have been shown to be very, very strong inducers of the GAR plus prion. So just quickly, this is SO2. This is what you use in a fermentation to control the initial population of non-Saccharomyces yeast and bacteria that are there. Generally, using SO2 results in quick, easy fermenting wines, but you tend to lose complexity because you get a lot of complexity from those wild yeasts and stuff that are in there at first. And so some winemakers really like to play around with adding sulfur late, or adding very small amounts of sulfur, or not adding sulfur at all. And if you've heard of natural wines, generally people are using less and less SO2. And sometimes it works and sometimes it doesn't. That's kind of up to you guys. Anyhow, back in the fall of 2013, I did a set of fermentations where I was testing the sort of the Prion yeast as well as the SO2 thing. So just show you guys real quick. The green line here is a wild type yeast that had SO2 added. So this is, let's call the green one a normal fermentation. The purple line was a GAR plus yeast that also had sulfur. So it started a little bit slower. It looked relatively normal depletion of sugar. But when it got down to sort of finishing fermentation off, it really struggled to finish fermentation. So this is saying there's probably about 2% sugar left and some, not as high alcohol, which essentially leaves you open for that later bloom of bacteria to come along and sort of spoil your wine afterwards. Yeah?
How early did you add the SO2?
So the SO2 was in the juice before we inoculated anything, yeah. So our winemaker put that in for me before I came down to the winery. And so the two fermentations without SO2, the GAR minus one, it had a little trouble getting started, but once it got going, it had relatively normal fermentation kinetics, and it finished dry. So it was in many ways sort of indiscernible from the one without sulfur, or the one with sulfur. The GAR plus without sulfur, however, really this one lagged almost like a full week before it got going. It had affected fermentation kinetics the whole time, and it really struggled to finish. So this was definitely what we call a problem fermentation, right? It smelled weird. I looked in it. There's lots of bacteria. But instead of just having me use my eyeballs to look at what was in there, we decided to do some DNA microbiome sequencing on it. So this is, again, sort of complicated data, but the thing I really want to just draw your guys' attention to was in the SO2 conditions, nothing really significant was different between the two metabolic states of the Prion minus and Prion plus. However, in the condition with no sulfur, the GAR plus yeast really struggled to dominate, and there was a huge bloom of some lactic acid bacteria that occurred sort of mid-fermentation and is very indicative of essentially a problem or a stuck fermentation. And you could smell this in the volatile profile of this wine. This wine was really like gross and buttery and did not smell great and did not taste good. Even a couple years out, it tastes pretty bad. So I would certainly not recommend this as something to do intentionally. So just kind of go back and overview my model here. The idea is that there's bacteria in the environment. They're spitting out some sort of small molecule effector, which we think we may have finally figured out what it is, but I don't want to tell you guys yet because we're still trying to figure that out. Essentially, the bacteria are putting stress on the yeast. The yeast are perceiving this stress, and as a way of sort of adapting to that stress, they're using molecular chaperones to refold the PMA1 proton pump into the GAR plus prion. And this is a heritable form of learning that when cells will replicate, they'll pass on this protein, the differentially folded protein within their membrane, passing on this phenotype of being able to use alternative carbon sources in the presence of glucose. From a winemaker's perspective, this is bad. However, from a yeast perspective, this is a great idea. They're sensing that there's competition in their environment, and they've suddenly figured out a different way of running their metabolism where they're less dominant of their microbial partners, but they have increased survivability. So we've seen that they have longer lifetimes, chronological lifetimes, as well as they survive longer in fermentation. And they're able to use different carbon substrates even when glucose is present. So they're sort of diversifying their metabolic strategies by doing that. They're no longer reliant on just glucose because they'll have competition for glucose if there's a lot of bacteria around. And they also gain some potentially increased resistance to certain toxic substrates and compounds that other bacteria and stuff spit out. So it sort of makes sense from a yeast perspective why they would maintain this prion. So just a couple closing thoughts on wine. I didn't mean to get quite so sciencey with you guys there, but we got through it. Cleanliness of your winery certainly makes a difference. Sanitation makes a big difference. Starting with sound fruit is a huge thing. You know, when people want to make natural wines, you want to do it with really, really nice fruit, because if you have crappy fruit that's all damaged and you bring it into your winery and don't add SO2, you will definitely have a problem. Enological decisions make a big difference. Choosing what kind of yeast you're using, choosing if you want to add nutrients or not, choosing the size of your fermentation vessel, how many pump overs, how much mixing you do, all these things can affect your microbiome of the wine and really affect the sort of long-term stability, flavor, and organoleptic properties of your wine. And yeah, the microbes that are in there really do matter. You know, beer is very much driven by the type of yeast you use, but wine is too. And I think the younger the wine is, the more sort of microbial character you get. But even long-term, even something that's been in a bottle for 20 years can still feel the effects of you know, something that happened right at the beginning of that wine's life. And you know, people ask me all the time when I tell them I work on wine, they're like, "Oh, what's your favorite? You know, what should I be drinking?" I say, "Well, the best wine is your favorite wine, right?" I don't love 2 Buck Chuck anymore, but if someone's into it, I say drink it and drink it proudly. You know, buy and drink what you like. And you know, my advice would be try as many wines as you can, figure out what you do like, and then you know, drink that. And if my favorite thing is sort of figure out a region that you pick, some grapes you like, and you can always sort of fall back on that. I happen to like Tempranos from Spain, and I really like Pinots from Santa Cruz and Russian River, and you know, a couple of Southern Rhone. Love the Southern Rhone. That's great. Good stuff. Anyhow. Thank you guys for listening. If anyone's interested in learning more, there's some publications. We have sort of a website. Inside Science TV made a video with us like a year ago, so it's kind of a wonky video, but it's on YouTube if you want to check it out, and or you can email me. But thank you for listening, and thanks to the Masson Lab for coming by. Cheers. You first. You had your hand up, and then.
So obviously you have to understand the science. have sort of practical assistance for winemakers where they see, it doesn't look like fermentation's going right, but you have like a gene sequencer right there with the wine, and go, okay, let's say we got some GAR plus, so we need to do this or that.
Yeah,
is
so
that
actually,
kind of where you think things are going?
Yeah, totally. In fact, there's ETS is the company that does sort of real-time microbiome sequencing stuff for people. So people will send samples of their fermentations off to ETS. They'll run a bunch of primers with it, and they'll tell them, oh, you have this mixture of different lactics and yeast. At the moment, there's no easy DNA method to detect the GAR plus Prion. So everyone in the industry, as soon as they heard this research, started scrambling to try to figure out how do I detect it. And so people started selling services to detect GAR before they actually even knew what it was, which speaks something to the industry itself. I've been working on this for 7 years, and I still can't entirely tell you with full confidence, to look at a sample of wine and tell you whether or not there's GAR plus yeast in it. We've seen situations where we put in yeast or bacteria that can induce GAR plus, and then pulled yeast out of those problem fermentations that in fact were GAR plus, according to our metrics. I think they're working on it. We have a patent with MIT, and I know they've been talking quite a bit to MIT about trying to commercialize this, but I don't think they have a real method figured out yet. But that is the eventual goal. What I want to do is end up going to a yeast company and helping develop yeast strains that will be resistant to going GAR for various genetic reasons. And there's also potential too that GAR changes the metabolism of the cell, but doesn't always massively impact fermentation. Sometimes it impacts fermentation just a little bit. So there's a chance that you could actually use these GAR plus yeast as a winemaking tool to potentially give you a different aroma characteristic from your wine. So there's sort of two cool ways to think about this, is that we can try to harness what the yeast are doing biologically inherently and make it work for us, or let's just avoid that altogether and sweep it under the carpet. So yeah.
Yes, so is there any work on what we might call the industrial internet of farming? All right, so sensors in the vineyards measuring, the instruments already exist commercially, but then the data analysis and integration, if you will, all the way down to your chemistry?
Yeah, absolutely, actually. There's a couple of people in the department doing that. I forget, Jerry, do you remember the fancy name for when people do the vineyard stuff? I don't know. They have all sorts of crazy sensors in the vineyard, and they can overlay like soil maps and rain maps and all the different types of maps. As like, I'm not a stats person. That's way above me, but it's cool. They totally do that. And there's actually a new faculty member, Ron Runenbaum, who's come in, and he's trying to define what terroir is, because terroir is such kind of like a whatever thing that nobody understands. That they talk about it a lot. So he's doing I think it's going to be a 15-year project where he's got the same blocks of Pinot Noir, and he's going to harvest across 15 different years, and he's tracking rainfall, tracking soil composition, tracking microbiome, all sorts of stuff. And he's trying to say, from these 15 different sites, given all this wine chemistry you can do, given all these other sort of metrics and things we can follow, what is defining these wines as giving the characteristic from each site? So I think that work is happening, and it's super interesting. If you ever, you can just check out, I think it's Wine Server at UC Davis, something like that, and that's our department website has a ton of stuff like that. So yeah, I'm not doing that, but absolutely people are using those sensors and that sort of next gen technology stuff to get there. So. Cool. Thanks, guys.